In the today's session, we are going to discuss the source of herbs from the chapter Herbs as Raw Material. In this session, we are going to discuss the various sources of herbs. These sources are either we are getting the herbs from the wild source, or we are getting the herb from the cultivated source, or the modern scientific techniques. If you can see the picture, see all the images which I have given on the slide. Very first one, there are some herbs are there, and many more medicinal plants we are getting from the wild source. If you can see the second picture, there is a cultivated aloe is shown, and definitely we will get some medicinal plants from the cultivated source, or the combination of the cultivated source along with the modern scientific technique. And the third source of herbal raw material, that is the modern scientific technique. So let us see the source of herbs, that is from wild source, from cultivated source, and the modern scientific technique. Move towards the very first point, that is the wild source. So wild source, the plants are obtained from the wild source and grow themselves without any type of care at unutilized land such as a forest, plains, river banks, and etc., etc. So what is the wild source? Maybe in the forest, maybe in the plains, or maybe in the river banks. Some plants are going to be grown themselves, and from that we are getting the source of these herbs, isn't it? Here we are not taking any type of care. Whether we are taking any care, suppose that the plant which is grown themselves in the forest, whether we are taking any special care? No, nothing else, isn't it? The care is being taken by the nature. The wild plants also have reasonable active constituents, and sometimes the new varieties produced in the nature. Sometimes in the nature itself, they are having the ability to produce new varieties, isn't it? And these plants grow under the favorable conditions in the natural habitat. So in this way, we will get some herbs from the wild source. So what are the advantages of the wild source for getting the as a raw material of herb? So very first one, it is economical. Whether there is any kind of investment in the growing of plants, there is no any investment. It's right. There is a zero investment. So it's economical. Whether there is a cost of land, whether we have to pay the cost of land, no, because the plants are present in the forest, plants are present in the plains, plants are present in the river banks, isn't it? So there is no need to pay the heavy money for the land. The third advantage, there is no need to take care of the plant. Whether we are taking a special care, no, isn't it? That is the third advantage of wild source. Fourth advantage, there is no requirement of any fertilizer. Whether we are going for the pest and pest management, in the case of if the plant is grown in the forest, no pest management is there, isn't it? There is no need to pay the heavy money for the pest management. Like when we are growing the grapes, we require heavy money for the pests and their management, isn't it? Then no irrigation is required. So these are the some points which needs to be considered under the advantages. That is, whatever the wild source is there, it is economical. There is no cost of land, no caring, then there is no need of the fertilizers, and no irrigation. Along with this, it is a less time consuming and decreased cost of labor. As we are not taking the care, so automatically the cost incurred on the labor, it is going to be decreased. So these are the certain advantages of the wild source. Let us move towards the disadvantages of wild source. Like advantages, there are also some disadvantages to these wild sources. So very first disadvantage is that the quality of plants cannot be predicted. Whatever the plant grown in the forest or in the wild, it is not possible to control, it is not possible to predict the quality of plants. The second disadvantage, that is a non-uniformity in growth and yielding characteristics. It is not possible to get the proper yield for the wild source plants or wild herbs, and also there is a non-uniformity. It is not possible to apply the modern scientific techniques. Say, for example, plant tissue culture. Whether it is possible to utilize the plant tissue culture in the wild sources? No. Hybridization, it is not possible. If nature is having a capability to hybridize the things, we will get some hybrid products also in the nature itself, isn't it? But as such, it is not possible to apply certain modern scientific techniques to the wild source plants. Then the last disadvantage, and which is very important one, there is a depletion of raw materials from the wild source. Automatically, if every time we are cutting the plants, we are collecting the plants, leaves, flowers, seeds, whatever the requirements from the forest continuously, automatically the things are going to be depleted unless and until we are not cultivating it in the forest, isn't it? If you can see, I have shown here one image, these are some of the medicinal plants which grown in the Himalayas, and definitely all these plants have the wild source. Means we have to collect all these things from the wild sources only, right? So this is about the wild source. So we have discussed what is the wild source, how the things are being collected, how it is growth, then the advantages of wild source and the disadvantages of wild source. Okay, let us move towards the cultivated source. So the cultivated source, the plants are obtained from the cultivated source also. See the figure, how the aloe vera is planted and cultivated. So that is again yet another source to get the herbal plants. And these plants are grown with a proper care by the human being. So there are some plots are there, and we are taking a proper care, pest management and each and everything, the use of fertilizers, use of water, and so on. And these herbs are going to be grown. The care is taken towards the soil, towards the climate, towards the rainfall, towards the irrigation, towards the time of sowing, and collection, altitude, temperature, fertilizers, manures, pesticides, seeds, and so on. So everywhere we are taking the care of the plant. We are selecting the proper soil. We are selecting the proper source of water. We are selecting the proper fertilizers for the plant. We are selecting the proper collection methodology. We are selecting the proper pesticides for it, isn't it? And from that source, we are getting the certain herbal materials, isn't it? That is from the cultivated source. Let us see the advantages of cultivated source. Already we have seen the disadvantages of wild source. So when we are comparing it with the cultivated source are having the much more advantages. And very first advantage is that the quality and purity is ensured, along with a better yield and more profit. That is the first advantage of the cultivated source. The second advantage, that is the application of modern scientific technique is possible, like PTC, plant tissue culture technique, and hybridization, and so on. The crop planning. Here in the case of cultivated source, it is possible to plan the crops to ensure the regular supply of raw material to the industry year on year basis. Suppose that yearly XYZ company requires the 1 metric ton of the aloe vera. It is possible from cultivated source to get the 1 metric ton of the aloe. Then application of scientific techniques like a tissue culture technique, then genetic engineering, hybridization, and germplasm, these are possible. Along with, if we are cultivating the medicinal plants or these herbs, automatically in a nearby area, there is a industrial growth, right? If you can see, take an example of sugarcane. There are so many sugarcane industries are available nearby the sugarcane cultivation, isn't it? Similarly, if we are cultivating the aloe vera, definitely aloe vera industries, herbal industries are going to be grown in a specific area. And that is the industrial growth. So these are the some of the advantages of the cultivated source. So this is about cultivated source. Let us move towards the modern scientific techniques. There are some modern scientific techniques are there which are widely used in as a source of herbs. Very first one, that is known as a plant tissue culture technique, or it is also said to be the tissue culture. Second one is a polyploidy. Then third one, that is a mutation. Fourth one, that is a hybridization. Fifth one, that is a genetic engineering. And the last one, that is a germplasm. We are going to discuss only the first 4 methods, that is a tissue culture, polyploidy, mutation, and hybridization. So very first one, that is the plant tissue culture. Maybe all of you are aware about the plant tissue culture. In vitro, we are growing some cells, and from that such cells, there is a generation of the plant, isn't it? If the things are being generated in vitro situation, we are maintaining the aseptic conditions. Definitely, it is a rich with the active constituent. If the medicinal plants are grown by using the concept of plant tissue culture technique. So PTC is in vitro cultivation of plant cells or a tissue under the aseptic and controlled environmental condition in a liquid or on a semi-solid when defined nutrient mediums for the production of primary and secondary metabolites or to regenerate the plants. The whole process requires a well-equipped culture laboratory and the nutrient medium. We are not going to discuss much more details of the PTC, but PTC, it is one of the techniques. See the figure, see the images which I have placed on the slide, how the plants are going to be grown in a vitro to get the some primary as well as the secondary metabolites from the medicinal plants. So this is about the tissue culture technique. So let us move towards the next modern scientific technique that is known as the polyploidy. So what is meant by the polyploidy? In this case, see simply what happens, suppose that there is a plant. Okay. And we are applying certain, or we are creating intentionally, we are creating certain situations, or we are utilizing certain chemicals. So automatically, if it is a diploid, it gets converted into the haploid. Suppose the size of leaves, it is a 1 mm. When we are applying the concepts of the polyploidy, the size of leaves are going to be increased from 1 mm to 5 mm. Polyploidy, it is an increase in a number of chromosomes in a certain tissues or entire plants in a multiples of the basic or a haploid number is known as the polyploid. Figure so that you will understand the things. See the normal diploid which is shown in the plant, and when in another part we have applied the colchicine, and see the difference between the second part and first part. See the size of leaves, see the size of fruit. When the colchicine is applied to terminal bud of the branch, the cells are tetraploid. There is 4n. Whatever the 2n is there, it gets converted into the 4n, and the leaves and fruits are larger, or we can say it as bigger. That is the polyploidy. You got the point? So polyploidy in plants is caused by the cell generation by using the physical agents and the chemical agents. So which are the physical agents? Like X-ray, centrifugation, change in temperature, shock, or the chemical agents like a colchicine. So that is the polyploidy. So what are the advantages of polyploidy? So very first advantage is that the plant is usually healthier, stronger, and larger than their diploid counterpart. See the figure. So whatever plant we have which is shown in the figure, after application of colchicine, its size is going to be increased. Size of leaves going to be increased. Size of fruit it is going to be increased, and the part becomes healthier. So automatically, whatever the active principles which are present in the specific part, it is going to be changed. It is going to be increased. Automatically, the yield is going to be increased for the specific plant part, isn't it? Then polyploidy plants contains larger flowers, pollen grains, and stomata. The phytoconstituents of some plants can be increased than the counterpart of a diploid plant. So here in the case of herbs or medicinal plant, the phytoconstituent, amount of phytoconstituent in polyploidy part, it is going to be increased. And that is the main use of a polyploidy in the case of source of herb. The main disadvantage of polyploid, that is the effect of polyploidy, is not generally predicted. It is not possible to predict its effect, isn't it? Always it is not possible to get the healthier plant, isn't it? That is the main thing when we are applying the concept of polyploidy. Let us move towards the mutation. So mutation, it is a change in nucleotide sequence of a gene which forms new genetic trait or changed genotype. A cell or organism which shows the effect of mutation, it is called as a mutant, and the agent which causes the mutation is known as the mutagenic agents. This mutation, it is furthermore divided into the three types. Very first one is artificial, second one is the radiation, third one is a chemical. So mutation, it is a change in nucleotide sequence of a gene which forms new genetic trait or change the genotype. Three types of mutations are there. Very first one is artificial, second one is the radiation, and third one is the chemical. So artificial mutation, it is induced artificially by exposing the plant to the abnormal environments such as the radiations, temperatures, or the chemicals. And radiation mutation, it is induced by the electromagnetic waves of a short wavelength, for example, UV, beta rays, or the gamma rays. And chemical mutation, it is induced by the chemical mutagens like a nitrogen oxide, formaldehyde, or nitrous acid. So these are the three ways by which the mutation in the plants takes place, and which will act as a source of herb, source of a phytoconstituents. The main advantage of mutation, that is it increases the phytoconstituents of certain plants. Let us move towards the hybridization. If you can see the figure, you will get the idea about what is meant by hybridization. We have the first two plants, A with a blue color, maybe B with a pink color. If we have hybridized these two plants, what will happen? Either we will get the C or D. The plant with a two-color flower, so which is shown in the C figure and D figure, right? If you can see the C figure, which comprises only the one flower with a two color. If you can see the D figure, one plant with a two colored two flowers, isn't it? That is nothing but a hybridization. So hybridization is a natural or artificial method in which union or crossing of two genetically different plants occurs. As a result, individual produce is known as a hybrid. So if we are uniting or crossing two different genetically plants, whatever the product we are getting, that is known as the hybrid, and process is known as the hybridization. So hybridization does not modify genetic contents of organism. However, it produces the new combination of genes. There are certain advantages of hybridization. That is, the desirable characters of two different plants can be obtained in a single hybrid plant, and the phytoconstituents of some plants can be increased in hybrid plant. So automatically the yield of phytoconstituents, it is going to be increased in the case of when we are hybridizing the things or two varieties. Let us move towards the further table. Very first table I have created for a source of herb, which shows the source, significance, and the example. If you can see the table, very first one is a plant. So plants which are widely available, large structural diversity, sustainable, renewable, and cost-effective source for these herbal raw materials. Say, for example, digitalis, cinchona, ephedra, senna, aloe, rhubarb, and raulfia. The second one is the animal. Definitely, some animals they are going to provide the important drugs. They are going to provide the enzymes from ancient times. If you can see the honey, then beeswax, wool fat, cantharidin, and the gelatin. Maybe you are aware about the source of gelatin from the porcine and so on. Then the marine source, that's the marine, huge source of many potential drugs, which are we are getting from the marine source like algae, some plants and animal. Say for example, agar, algae, and certain antibiotics. From mineral source, there are certain naturally occurring minerals are there, like salts, oxides are used as a medicine from ancient time, and they are stable and effective on various diseases. For example, zinc oxide, calamine, and shilajit. Maybe you have seen the use of zinc oxide and calamine in certain cosmetic preparations like a calamine lotion or zinc oxide paste. Microorganisms which are discovered from the microorganisms and which are widely used to get the certain antibiotics, enzymes, and vitamins. Then modern approaches, for example, PTC, plant tissue culture, biotechnological approaches, which will give us the insulin, hormone, secondary metabolites, then antibiotics, certain produced by the fermentation. So these we are getting from the modern approaches. The next table, which is associated with the drug isolated from the natural products. I have listed here the certain drugs which have been isolated from the natural products. For example, the atropine, which we are getting from the belladonna, and the use of atropine is the anticholinergic, right? The second one, that is the codeine, which is obtained from the opium, and it is having an analgesic and antitussive properties. Furthermore, the digoxin, which is obtained from the Digitalis lanata or purpura, and which is used as a cardiotonic. Then ephedrine, which is obtained from the ephedra, which is a bronchodilator. Then phenocide A and B, which is obtained from the senna, and it is used as a laxative in the constipation. Then vinblastine and vincristine, which is obtained from the vinca, and which is used as a anti-cancer. Apart from this, again I have made one table by considering the parts of plants. So let us see how the things are going to be isolated from the parts of plants. For example, the roots. We are using the roots of shatavari, raulfia, aconite, jalap, and ashwagandha. There are certain drugs are there which are being collected from the woods. For example, sandalwood, cassia wood, and red sandalwood. Then source as from the bark. For example, cinchona, cinnamon, kurchi, and oak. Then rhizomes like a ginger, valerian, turmeric, and podophyllum. The flowers are being used from the saffron, the tea, and artemisia. Artemisia, it is being used for the malaria, which contains the artemisinin. Then the fruits from fennel, coriander, amla, and gokhru, isn't it? Then the leaves like a senna, digitalis, vasaka, and eucalyptus. Maybe eucalyptus comprises the collateral. Senna contains phenocide A and B, glycosides. Digitalis contains the digitoxins, which is source of digoxin. Then seeds like a nux vomica, isabgol, and mustard. Whole entire plant or entire drug, it is used from certain plants like a neem, brahmi, vinca, and belladonna. Then dried lattices from the opium and papain. Papain, it is going to be used as an enzyme. Then resins, say for example, balsam of tolu, then myrrh, asafoetida, benzoin, and guggul. The gums like acacia, travacanth, and guar gum. Dried juices which are obtained from the aloe, quinoa and red gum, and dried extracts from the gelatin, catechu, and the agar. Last table, which is related with the unorganized drug, along with their description and example. So, the first category, that is known as the gums. These are the pathological products produced by the plants as a response to injury. If we are giving any injury to the plant, which is going to produce, which is going to extrude certain things, that is the gum. They are hydrocolloids and may be anionic or non-ionic polysaccharides. On hydrolysis, yields sugars and uronic acid, which forms salts with calcium and magnesium. It is soluble or partially soluble in water, insoluble in alcohol, and in most of organic solvents, for example, gum acacia, tragacar, gum ghati, guar gum. Mucilages, these are again physiological products produced by the plant as a normal metabolism. It is produced inside the cells of plant. Mucilages are esters of sulfuric acid, wherein the ester group is polysaccharide complex. Mucilages from slimy masses with water but do not dissolve. For example, isabgol, squill, and agar. Extracts, these are the solid or semi-solid masses obtained by the extraction of drug either by water or other solvents, for example, agar, sodium alginate, catechu, gelatin. Resin, oleoresin, oleogum resin, they are solid, liquid, semi-viscous material, clear, translucent, yellow or brown material, insoluble in water, soluble in most of the organic solvents along with the alcohol and chloroform. The examples of resins like porphyrin, colophony, oleoresin, for example, capsicum, ginger, balsam, tolu, benzoin, oleogum resin like myrrh and asafoetida. Then lattices, products contained in a special secretory tissues of certain plants. It is usually a white suspension. Oil globules are suspended. It may contain alkaloid, protein, sugar, mineral, and alkaloid salts in a true solution, whereas gums, starch, and resins in the suspended form. For example, rubber, opium, and papain. The juices, which are obtained from fresh leaves, fruit, stems of the trees by cutting and crushing. For example, aloe and quinoa. So in the today's session, in brief, we have discussed the source of herb, which is obtained from wild, cultivated, and modern scientific technique. Along with this, also we have discussed in brief all the drugs along with their source and the use. So herewith I declare that the today's session is over. Thank you very much.